Mendel have to offer? So you're going back a bit. So Gregor Mendel, this turns out he was a monk. He was in fact quite a successful monk. Tried to be a teacher, trained and took the exams, I think twice, failed them both times, then gave up on being a teacher just as well, really, because it turns out that his skills were elsewhere. He was really successful as a monk, though. He became abbot of the monastery, and this is in the former, I think it was former Hungary, Austria, I think it was at the time. So he spent most of his life working in a monastery, and that's where he did his research on genetics. So he his work was, unfortunately, though, kind of largely unknown until after he died, and then it was discovered by lots of people. So what did he do? Well, he was a keen gardener, and he got really interested in, studying the differences in and what was behind the differences in the characteristics of the plants that he was growing. So he started to think up experiments that he could do to try and figure out what is going on, and what are these patterns? Are there patterns, and what's the possible explanation for how it is that characteristics are passed on from generation to generation. People kind of knew that was the case. It's kind of obvious, right? Children kind of look like their parents, and if lots of people were breeding plants and animals, and they kind of had, but they had kind of mixed up ideas about how traits, how characteristics were passed on from generation to generation. They had no idea. Remember, Mendel had no idea about genes, chromosomes, didn't know anything about mitosis or meiosis, or all of that stuff was unknown to everybody at the time. Okay, didn't know that DNA was, even the existence of DNA. That wasn't until like 100 years later when Watson and Crick figured out that not just the structure of DNA, but the DNA was the molecule that somehow carried the information from one generation to the next in the gametes, right? So he chose peas as his model organism for his experiments, and he did a ridiculous number of experiments. And you'll see that he was so dedicated. He didn't just do experiments; he did them. He did experiments that were just painstakingly detailed and repeated over and over, hundreds and thousands of times, to make sure that the data that he was getting was robust, right? So he was, in a lot of ways, he was really like an ace scientist, right? So he chose peas. Why did he choose peas? Really easy to grow, very short life cycle. So you could, grow a plant from a seed. And by the way, a pea is the seed for a pea plant. So if you take a pea from a pea plant, from the pod, and you plant it into the soil, it'll grow into a pea plant, right? But that doesn't take very long. You can grow a pea plant in just a few weeks. So he was able to do lots of experiments because the peas were so quick and easy to grow. It also meant they didn't take up too much space, so he could grow a massive number of plants. And then it meant that he was able to do, have huge numbers for his data, right? There was lots and lots of data, basically. So we chose because they can be self-pollinated. I'll show you what that means in a second. Now, he kind of knew enough. He realized early on that the secret to success was to be able to keep true breeding, groups of true breeding strains. Now, what true breeders are, Alex just explained. Purebred, you might have heard of purebred or true breeding. What this means is that a true breeding, imagine if you have a true breeding plant that and the characteristic that it's true breeding for. So you talk about true breeding for a particular characteristic, let's say tall versus dwarf, right? So tall pea plants, he kept population of tall plants that if you took any random member of this group of tall true breeding plants and you cross them with another member of the true breeding tall plants, you would always only get tall offspring, right? That's what the true breeding meant. If you took true breeding dwarf plants from his group of true breeding dwarfs over in the other part of his greenhouse or whatever, and if you crossed any of those true breeding dwarfs with another true breeding dwarf, you would only get small dwarf offspring, right? So he had developed these true breeding strains, not just tall and dwarf. He had all kinds of different, he tracked all kinds of different characteristics. We'll see in a second. But what he realized, so look at this. This is the kind of stuff that he looked at. He looked at, different types of seed coat color or pod shape, pod color, the position of the flowers, the tall or small. So colors of flowers, all kinds of stuff, right? And he did experiments on them. So I think I've been through this. He kept lots of huge numbers of plants, and he was very meticulous about recording his results. So he started doing experiments. So he took one of his true breeding plants, his true breeding pea plants. He picked a true breeding purple one to start with. So these were purple plants that if you cross them with another true breeding purple, you'd only ever get purple offspring, right? But he had another group of white ones, very similar in every way except just white instead of purple flowers. And again, if you took those white flowered pea plants and you cross them with another white, similar true breeding white flowered pea, you'd only get white offspring back. So he took these two populations. He took one purple one and one white one. No, not just one. He did this hundreds and hundreds of times. I'll show you in a second. But the experiment goes like this. Take a purple one, take a white one, cross them. And by the way you cross, this was the very handy thing. Another reason why he chose pea plants was because you can just get a paintbrush. This is what he spent his days doing. Take the pollen off the one plant, off the male part, the stamens, the anthers, right, of the purple plant, and transfer them over to the white plant. You could do the other way around as well, it didn't really matter. But if you crossed one with the other, and then you collected the peas, which were the seeds, right? So here's the pea pods, and those seeds are the fertilized ovules, right? They're the result of the fusion of the gametes that are in the pollen, the gametes that are in the carpel inside in the ovule, in the ovule. Once they fuse, you get these seeds forming. And when he planted those seeds, he found that every single time, 100% of the time, he got purple flowering plants back. So that was a puzzle, right? What's happened to, where'd the white go? Right? So all the time, they were true breeding purple crossed with true breeding white, 100% purple offspring. So that was a bit strange. So he did a second experiment. He took those first generation, what we call the F1 purples, right? These are the product of the first cross that he just did. These are called F1 because F standing for filial, so filial like brothers and sisters. So he took the F1 hybrids. They were the result of the cross between the purple and the whites, right? But they're all purple. He took them and he crossed one with themselves, basically, with another F1, right? You can actually do self-pollinating with peas. So instead of crossing with another similar F1 hybrid, he just was able to take the pollen from the anthers in a flower, and probably not on exactly the same flower, but on the same plant, he was able to put them on the, brush them onto the stigma of another flower on the same plant, right? So self-pollinating, not the same flower. That's just kind of like for our story, for our cartoon. So what did he find? So he found that now in the next generation, when he took those peas from that cross, you get purple and the white pops back up again. So that was strange in itself. But what was particularly, this was the genius of Mendel, right, was that he found that no matter what trait he did, flower color, pea shape, pea color, plant height, whatever he did, he always, and he because he did such huge numbers, he was able to see this ratio coming out again and again and again. He was able to see almost exactly every time, he was able to see that it was 3:1. So he didn't just do this, like I said, with, let's skip that. He didn't do it with flower color. He did it with all of these different things. Round versus wrinkled peas, right? And the ratio, look at the numbers. This is a guy who's counting 5,474 round peas of a day and 1,850 wrinkled ones to get the ratio of 2.96 is to 1 or 3.01 is to 1. So he kept seeing, only because he was just so meticulous and detailed and determined, and did such huge numbers of experiments, he was able to see, okay, this is something going on here. 3:1, this is the magic number. Why is it 3:1? Was his question. Why is it that first of all, that the white disappears in the first generation and then pops up again? Not just that it pops up, but it pops up exactly 25% of the time in the progeny, in the offspring, in the second generation, the F2 generation. So that was his puzzle. He spent a long time, first of all, getting the data, but then the genius was really, how did he explain this? So this is how he explained it. Now, there are two laws in this chapter, Mendel's two laws, and they're really, really crucial. You know, if you're going to do nothing else, learn the two laws, right? But they really explain, they help you to explain how to do the crosses that we're going to be doing over the next while. So here's his first law. And I'm going to do these hand motions because this is what a biology taught me one time, it helped me as well. So when you're in the exam hall in June, look around you. I bet you're going to see a couple of people doing this kind of thing, and they're trying to remember what Mendel's first law is. So Mendel said that every characteristic is governed by a pair of factors. He didn't know there were genes. He didn't know about, like I said, chromosomes and genes. But from the results of the data, he said this is the only explanation that makes sense of all this. He said every characteristic is governed by a pair of factors, is what he called them. Didn't know where they were, didn't know what they were. He just knew that they must exist. So a pair of factors govern a characteristic, like flower color or pea color or plant height or whatever, right? Pair of factors. That these factors separate during gamete formation, so that each gamete receives one of the pair of factors, right? And then we know, and he knew then that this wasn't really the important bit of the law, that those factors, they reestablish in a pair at fertilization, right? Once the two gametes fuse again, then the pairs are reestablished. Like in humans, peas are not that different to humans. I know we like to think we're different, but we're not that different. So again, every characteristic is governed by a pair of factors, that the factors separate during gamete formation, so that each gamete receives one of a pair of factors, right? Then they come together again in fertilization. That's Mendel's first law, and it helps make sense of the experiments that we just saw. So here now, we can look under the hood as Mendel figured out this is what must be going on. So if you look, because the purple ones, that purple color is governed by a pair of factors, right? In this case, they happen to be both the same. They're both what we label them big P. Big P stands for purple. We use a capital P for purple because purple is the one that seems to be the one that's the dominant trait. We'll talk about dominant and recessive in a minute, right? White is this recessive trait, the one that gets, I don't want to say the word masked because that's not what's going on, right? So small p for white, we choose P because purple, P for purple is the dominant characteristic. So when you take these two true breeding, and the reason that they're true breeding now you can see is because like the purple plant only contains purple genes, whereas the white plant only contains white genes, right? So if you take those white plants and you cross them with other similar white plants, well, you're only ever going to get white because that's all there is. In the purple, similar, you take purple ones, you cross them with other similar true breeding purples, you're only ever going to get purple back because there's just purple genes available, right, in the gametes. No matter what gametes are formed, the gametes are just going to contain purple genes or white genes for the white plants. Okay, but when these pairs of factors separate during gamete formation, so that each gamete receives only one of the pair of factors. Now, in the case of the first cross, you can see, well, there's only one type of gamete produced by the purple plants. That's the purple gamete, and there's a white gamete produced by the white plants. Because so they're all, there isn't going to be any variation in the gametes, right? When you put those gametes together, when the eggs, this is what we call a Punnett square. I'm sure you're familiar with these. And there's no need really to do a Punnett square for this one because you can see straight away there is only one combination of gametes here, one combination of different versions of the genes, right? And it's big P, small p. That's all there is. There's all the way along. There can only ever be, all that's possible is big P meets small p, and you get a purple offspring plant. That's why then in the F1 generation it was 100% purple. This makes sense of his first experiment. But even more than that, his first law made sense of the second experiment as well, right? So when you took those purple plants, and by the way, those purple plants look exactly like the other purple plants, the true breeders. You can't tell by looking at them; they look just the same. Well, in the first generation, Mendel realized, they're not the same though. When you look under they can't be the same. They must have a purple gene and a white gene. I shouldn't call these purple genes and white genes. Let's use the word that is the correct word for them. So there's a flower color gene, right? And there's different versions of that gene. You can have a purple version of it and you can have a white version of it. We call those versions alleles. So there's a purple allele and there's a white allele. And in these F1 hybrids, there was one of each, but they still were purple. And so what he realized was, but that's because these different versions, in this case, and in lots of cases in genes, there's a dominant version, and then which is the one that kind of comes through, and then there's a recessive version which is in the background and doesn't get expressed. You don't see it in the physical plant. It's there, but it's not being what we call expressed, right? So now, when you do the Mendel's first law, again, gametes are formed when that each characteristic is governed by a pair of factors which separate during gamete formation, so that each gamete receives only one of the pair. Now you can see one type of gamete is going to have purple genes, purple alleles, and the other gamete is going to have white alleles. And it's the same for the other plant. These are identical plants, right? Now, when you do the Punnett square, and remember how these come together, the fertilization events are equally likely. It's just as likely that you have big P combining with small p as big P with another big P, right? So this is what Mendel figured out, is that when you do this working out, it fully explains why you get a ratio of 3 purples to 1 white. So his explanation absolutely perfectly fitted the data, and he had a serious amount of data. So this was his, this was a triumph. This was him using, and it was like, this is just such a great example of how to do good science, right?